to New York City Atheists Live on Tape, brought to you by New York City Atheists Incorporated. I'm your host, Dennis Horvitz, the David Letterman of the Hopelessly Damned. Without further ado, please welcome Richard L. Borowsky, Professor of Biology at NYU. We're going to find out why flying fish are lying.
Oh, I'm not going to talk about any of that.
We have evolved beyond it.
So in the last couple of minutes, I've decided that, actually half an hour or so, I've decided that this is really a tough room. It's more shaped like a bone residual debate as to whether eyes are selected against in the dark or not having a function. There's natural selection will not be policing and screening new mutations that come along. So new mutations might come along, not be selected for, but just by chance increase in frequency in the population, and structure tends to run downhill if it's not tended. The metaphor I used before was the metaphor of the untended house. If I move away from my house for 20 years and nobody's watching it, I mean, I know for sure when I come back there'll be window panes broken, there'll be leaks in the pipes, there'll be squirrels in the roof, there'll be there are squirrels in the roof, but what I don't know is where these window panes will be. I don't know which tiles or shingles will be pulled up by the squirrels and so forth and so on. So if natural selection is not policing a phenotype, keeping it up to snuff, keeping it up to par because it's needed, in this case because it's not needed, then things can go downhill. That's called genetic drift as opposed to natural selection. So, major questions, I'm not sure I'll cover them directly, but if it is selection, how? Referencing Darwin's dilemma, I don't really understand it, he said. Mutation, is it random mutation, is it drift? I've explained it all. And one of the things that we're interested in as well is how many genes have been changed between being an eyed surface animal and a cave fish. Is it one gene that's knocked out, or 2 genes, or 10 genes? Before we started this research, nobody actually knew. Now we know it's probably on the order of 10, 12 genes per population. So I haven't any memory of why I put this in here. I assume that I'm going to show you fish from Tinaja, Pachon, and Molino, or something like that. Oh, yeah. So here's our surface fish. We know the polarity of change. Here's the Molino cave fish, which is an albino, as I mentioned. And here's one from a different cave, Tinaja cave. You can see a little bit of dark pigment in the mid-lateral line of that fish. These are actually not albinos, but they have greatly reduced numbers of melanophores, these pigment cells that are found in greater number in these guys. And when I hybridize them, what I obtain is a fish that in its pigmentation looks very much like the surface fish, maybe even darker than the surface fish, and very different than either of its parents. This is a phenomenon that's called complementation, and let me illustrate it with an example from human biology. Hearing disabled people tend to hang out with one another. I mean, they speak the same language. I can't do that, but they can understand one another with sign language. They tend to marry, as in all marriages, they tend to have children, and many times their children are also deaf. They're also hearing disabled. Sometimes, however, the child is able to hear. And that raises the question, is this guy the father of the child, or is somebody else the father of the child? And most of the time, he actually is the father of the child, 9 out of 10 times. And what it is she's deaf because gene A is bad. But gene B, which is also involved in hearing, is good. He's deaf because gene B is bad, but gene A is good. So he contributes in the sperm 1 good copy of A and a bad B. She contributes 1 good copy of B and a bad A. And the good copies of A and B are sufficient to make a hearing enabled child. And so that's complementation. The good qualities of one complement the bad of the other, and vice versa. And so here I have this diagrammatically for pigmentation. One of the fish, let's say Molino, has a good copy of A, but is homozygous. One copy from its mother, one copy from its father. Of B is little b, but it needs big B for dark pigmentation. It doesn't have it. Here, the fish from Team Naha is light in pigmentation. It has little a, and that's the deficiency. It needs big A, but it has big B. He contributes big A and little b. She contributes little a and big B, and the offspring is dark because it's complemented. That was a much simplified diagram. We now know that it's about a dozen factors in each population. But these dozen don't, I wish I had 3 hands, these dozen don't overlap with these dozen, and therefore you have this complementation. The complementation is also seen in vision, and so here's a 2-minute video illustrating this. Unfortunately, I can't play the soundtrack, but I can narrate it for you.
The Mexican tetra has 2 forms.
The Mexican tetra has 2 forms.
Cavefish are black, sterilized, and blind. Purebred cavefish are always blind.
I feel like I'm dowsing. I'm looking for the for the microphone. But the purebred cavefish always are blind. When you hybridize them, however, you can get fish that can see. So what we did is we tested them in this little apparatus with this cup with white and black barrel staves. You put the embryo, because we worked on embryo on fry, you put the fry in the bottom, you whirl the cup. If the animal can see, its eyes follow the stripes and snap back, follow and snap back. So this is a surface fish, very small fish, big eyes. You can see them flicking, and in this bubble and in the shadow on the bottom you can see the movement flicks. This is a cavefish with small eyes, and you can see the bubble is, the reflection in the bubble is turning. The eyes aren't moving at all. It's really blind. This is another cavefish. You can see the movement over there, but the eyes aren't moving. So he's blind. And then I'm going to show a hybrid between those 2 cavefish forms, and you'll notice that the eye spots are much larger. So eye spots are larger, and you can see them flicking as the surface fish eyes flick. So this is one generation of hybridization, one cave and another cave. Just as with pigmentation, these guys are blind because of changes in a certain set of genes. Those guys are blind because of changes in a different set of genes. In the hybrid, you get this complementation.
These experiments show that fish can be restored cavefish if you make the right genetic crosses. So learn more about this exciting.
So it's actually the same diagram that I showed before to explain restoration of pigment, in that it was picked up by Richard Dawkins. He really liked that story. And it was also picked up by a website called Creation Safaris. The creationists really like evolution in cavefish. Their argument is that only God can make something as complex as an eye, but random mutation can break down something that God has made and destroy it. So they're perfectly happy with the idea of regressive evolution. And in fact, their synopsis of my paper, I thought, was the best written and clearest of all the synopses that I read. It was really quite remarkable. And I know who I'm talking to, and I feel the same way myself, but it really quite amazed me. So, 3 conclusions, and I can shut up and take questions, or go. First, convergent populations or species can profitably be analyzed as replicates of evolutionary experiments. And I guess I haven't really shown that. It would be what I would talk about next if we had time for it, but maybe we don't. In cavefish, okay, this is the part that I haven't really explained to you. Direct selection is implicated in eye regression. And the third is that people can read into any story whatever they feel like. If you'll give me 3 more minutes, I will summarize what I would have shown you on those other slides.
Can you talk about leaving a direct selection?
Okay, if you give me 3.5 minutes, I'll show you. So, if at any genetic locus you hybridize a cavefish with a surface fish, in the F1 hybrid you have heterozygote. It's cave surface. It's gotten one gene from one parent, another copy of the gene from another parent, and the other copies, we use the term alleles. The alleles are different. So gene has actually 2 meanings. One is a place on a chromosome, a genetic locus. So we might talk about the eye color gene. It's here in the genome. The hair color gene, it's there in the genome. But at that eye color locus, you could have a gene for blue eyes or brown eyes or whatever. And that second meaning of the word gene we use allele for, different alleles. Then if you breed an F1 with an F1, you get a 1 to 2 to 1 ratio of homozygote, heterozygote, homozygote. If you test enough genetic loci throughout the genome, you can use the information from that experiment to put together a picture of the genome. So here's one of the linkage groups or chromosomes in that genome, and here's a gene called OCA2, which is the gene for albinism we've been able to show in those fish. And here's something called 218E, which is just an arbitrary designation. All of those numbers are arbitrary designations for loci that we can demonstrate. The whole map would be far more extensive. It would have 25 of these linkage groups. We have 23. They have 25. And given the map like this, we can ask, does it matter at 105B whether an individual is cave-cave, cave-surface, or surface-surface? So is there a difference in eye size between those 3? And most of the time, the answer is no. So when we look at gene 205E, the CC individuals, cave-cave at that locus, are medium, have medium-sized eyes. The heterozygotes have medium-sized eyes. The other homozygotes have medium-sized eyes. And what that means is that 205E has nothing to do with controlling the eye size differences. And it's not near anything in the genome that has anything to do with controlling those eye size differences. But when we look at 218E, these individuals in this class tend to have smaller eyes than those in the heterozygous class, which tend to have smaller eyes than those in the homozygous surface class. What that means is that near 218E, there's a gene that actually is controlling the development or the maintenance of the eyes. So something that's responsible. And so we use this sort of analysis to find about a dozen such places in the genome that really were important for eye size. And here's the answer to your question. When we did this analysis for eye size, every one of those places we found had the same polarity, in the sense that the surface-surface class, each one of these lines it denotes the trajectory for an individual gene. The surface-surface class was always bigger than the cave-clave class. But when we did the same analysis for number of melanophores, we found that most of them were in, what we call the right direction, but some of them had exactly the opposite polarity. And the difference between this pattern and that pattern is statistically significant, and we replicated it, the importance of replication, replicated it in an analysis of another cave population, so we're pretty sure that fate didn't play a trick on us. And the interpretation of this consistency is that natural selection is doing the job. Every time one of those mutations comes along, it increases the fitness of its holder, and it increases in frequency in the population. Whereas here, we view it as a case of genetic drift. If you don't have a melanophore cell there, you're going to have some other cell there. It really doesn't matter what cell you have there in your skin. If you're in the dark, you're not making melanin anyway, so it doesn't matter whether you have a melanophore cell there. There are several predictions that come from this. One of them is, since natural selection changes gene frequencies very quickly compared to genetic drift, we would predict that eye size decreases more rapidly in a cave population once it gets into a cave than the number of melanophores. We've looked at 6 other species of cave fish, and this prediction is borne out. The second part of the hypothesis is reason. Why should eyes be selected against in the dark? The brain is the most active tissue in your body, and the retina has a higher metabolic rate than any other part of the brain. It's an extremely expensive tissue. In us, 10% of our rods and cones are shed every day, so we renew the photoreceptor layer 36 times a year. Now, if you put me on limited rations and took out my eyes, the savings in energy probably wouldn't amount to very much, because I'm a mammal, I have a high basal metabolic rate. But fish, when they're not moving, have almost a 0 metabolic rate, and I wouldn't be surprised if the retina uses up maybe 1% or 2% of its daily energy budget. Testable hypothesis. I have a colleague in Scandinavia who's now doing those experiments on oxygen uptake of isolated retinas. So does that answer your question? And so I've spoken too long. I thank you for your patience, and I'll take any questions you have.
I have 2 good questions. One is one of the mutants found eyeless, the eyeless gene, or PAT6, I think it's called in vertebrates. That's one question. Second, as a fruit fly geneticist, I would like to ask if somebody has ever grown fruit flies for years and years and years in the dark. I suppose they have, but I haven't looked at the literature. I suspect you have. And have they lost their eyes? Okay, that's 2 questions.
So the first question was this master control gene, eyeless. It's called eyeless in Drosophila, PAC6 in other organisms. Has that been implicated? It was the first thing that we looked at. It has nothing to do with it, and these places during the or throughout the genome that we've identified, none of them are near where PAC6 maps.
Wow.
Yeah.
Wow.
So, first question. Second question, I don't know about experiments looking for eyeless condition or inducing eyeless condition in Drosophila in the dark.
Eric, I'll take that.
It would be a great thing to do.
Eric,
But one of the problems with doing an experiment like that is it takes 10 or 12 years, and you have to be publishing a couple of papers every year. And so that's something you do in your basement or not in your laboratory. Sorry.
Yes. Is this working yet? What does this all mean to us ordinary people? I mean, how does it affect us humans? And what does it mean to us? Why should we pay attention to this?
Especially if creationists say God created the earth, and we atheists want to be able to say, well, we have important evolutionary evidence that maybe it's not so.
Well, I'm not working for NYC Atheists, and this is not my main consideration. I find creationists extremely irritating, and I've had requests for information from them. Stephen Jay Gould, before he died, I asked him about this. He says, just don't even deal with these guys. There's absolutely nothing you can say, and I think there is nothing that you can say that will convert them. They've been converted. To me, the most amazing thing is that with probably 5,000 religions on the surface of the earth, how people can believe they have the great luck to be born into the one true religion. So that's a different story. In terms of intellectual excitement, I think that's fun. And so that's the value that I get out of it, and I hope it's a value, and I know that it's a value that's imparted to kids, because I maintain this great fish room with 7 different species of blind cavefish in it, and everybody who visits the lab, they bring by their kids. They really love it. So I think that has a value in and of itself. And I don't have to actually listen to atheists' interpretation of it. I think, so for example, one of the things that I've discovered is, you know how they say a shark can detect a drop of blood in the water a mile away? I don't know whether that's true. I've never seen any experiment along those lines. I mean, does somebody go out and cut himself and then wait? But I know that my fish, the surface fish, can detect amino acids, because I've done the experiment, amino acids in the water down to 10 to the minus 6 molar. I can translate that into real substances or real values that you can understand. My cavefish can detect amino acids in the water down to 10 to the minus 11 molar.
Wow.
So if you had 2 Olympic-sized swimming pools and you spat into one, and it was just pure amino, it's a vulgar illustration, but it makes the point, and it was pure amino acids in your saliva, and you stirred it, and then you took a cavefish and you dipped it into one, and then dipped it into the other pool, it could tell the difference. I don't believe that that's regressive in any way. That's a progressive change, and so I think there are elements of constructive evolution that the cavefish show. Finally, although it isn't what motivates me, the same genes that are causing loss of eyes in cavefish are the genes that are really necessary. There's no such thing as an eye loss gene. What there is the silencing or the corrupting of a gene that's necessary to build eyes or maintain eyes. Some of the phenotypes that we see are really reminiscent of human eye conditions like macular D, where we have a late age-related degeneration of the retina. And so if we could come up with a model for human eye diseases, I think that would be useful. Most of you are younger than I am, and some of you may actually be able to put your head down on the pillow at night and wake up 8 hours later. Don't work that way with me. I'm up 2 hours later worried about this, then I get back to sleep, and then I'm up at 4, I worry about that, and I get back to sleep. So the bouts of sleep that I have are many of short duration, and the difference in sleep patterns between the cavefish and the surface fish have to do with the number of bouts and duration. I know that NIH is very much interested in sleep disorders, so I think it's intellectually exciting. I think it has practical significance, and I'm really not out to try and convince people who believe otherwise that I'm right and they're wrong. Does that answer your question?
Yeah, thank you.
Can I just to kind of summarize, would it be possible for you to just kind of take us through the journey, say, of a sighted fish, and just kind of, at least maybe just schematically plot out the point at which its descendants are eyeless. A sighted fish swims into a cave. 100,000 years later, its descendants have no eyes.
Yeah.
Could you just kind of take us through that?
Okay, you just did.
Thank you very much, ladies and gentlemen. So, always glad to help.
So,
my office hours are...
there's this fish that shows up on a menu, tilapia. It's an African cichlid. And one of my ambitions is to die of old age without ever eating tilapia. About 25 years ago, the Department of Urban Ecology, I think the department has probably changed its name, in Mexico, decided that it had to feed the people. And what it did is it got large quantities of these tilapia fish and dumped them into streams and rivers all over Mexico. Amusingly enough, the fisheries department of the same government recognized that this was a form of pollution and would go out to these same streams and kill all the fish there to get rid of the tilapia. But the tilapia won. When I go into caves now, I find tilapia in the cave. That really means that if there's a cave and if there's a fish on the surface, within a short amount of time, that fish gets washed underground where either it can't survive or it can survive. If it can survive, then the population is maintained there. It's a radically different environment. And I really don't know the answer to the question, does it take 10,000 years, 100,000 years, 1,000,000 years? I know it can't take 5 million years because the fish got into this area from South America only about 2.2 million years. I know that the process has run its course in all of these separate different populations. So if I imagined that it took a long time, it took these 2 million years in order to reach this end state, I would predict that I would go into one cave and they wouldn't be halfway there, and another one they'd be very much there, and it would be all over the map, but it's not over the map, so I'm guessing that it takes a short time. That wasn't short for the answer, but does that answer it?
That's fine. I'm just going to, yes.
The question